Welcome to Medicalink. Don't forget to subscribe for more videos. Let's learn.
Hello friends, today our topic of discussion is urea cycle. The main goal of urea cycle is to eliminate ammonia from the body by converting it into urea. So the ammonia present in the body is taken to the liver where it gets converted into urea. Urea thus formed is taken to the kidney and it gets finally eliminated. So where does this ammonia comes from? In muscles, alanine combines with alpha ketoglutarate in the presence of the enzyme alanine amino transferase and gets converted into glutamate and pyruvate. This glutamate, it enters into the blood circulation and this glutamate will be taken to the liver. In the liver, this glutamate will undergo oxidative dehydrogenation to get converted into alpha ketoglutarate in the presence of the enzyme glutamate dehydrogenase. During this process, hydrogen ion will be removed, so the hydrogen will be picked up by NAD plus and will get converted into NADH and H plus. During the process, ammonia is released. This ammonia combines with hydrogen ion and gets converted into ammonium. This ammonium is more toxic and it is water insoluble. Now this ammonia will be taken into the blood circulation. So ammonia from the liver will go into the blood circulation. And in brain there are specialized cells known as astrocytes. These astrocytes will produce glutamine synthase. These astrocytes will produce glutamine synthase, and this glutamine synthase converts glutamate into glutamine, and this glutamine is osmotically active. This ammonia present in the blood will enter into the brain through cerebral circulation, and this ammonia will stimulate glutamine synthase to produce more glutamine. This glutamine is osmotically active, and at the same time this ammonia will incorporate into the cell to produce more of glycine. So ammonia is producing more of glycine and glutamine. This glycine and glutamine, they both are osmotically active. So what they do is they suck more water into the brain. So there will be accumulation of water inside the brain. This will result in cerebral edema. In cerebral edema, there is increase in intracranial pressure, which will result in brain herniation. Because of hyperammonemia, there will be cerebral edema, encephalopathy, and respiratory alkalosis. Ammonia can be generated to toxic level when there is certain condition like excessive protein breakdown. So when there is more protein in the diet, then this protein will start to break down, resulting in hyperammonemia. So this pathway can be prevented by pushing the ammonia into the urea cycle. The urea cycle will occur in the mitochondria and the cytosol. So there are 5 steps of cyclic process with 5 distinct enzymes. So the ammonia will enter into the mitochondria where it combines with carbon dioxide in the form of bicarbonate, and one molecule of ammonia combines with one molecule of carbon dioxide to get converted into carbamoyl phosphate in the presence of the enzyme carbamoyl phosphate synthase type 1, that is CPS. Now this carbamoyl phosphate will condense with ornithine in the presence of the enzyme ornithine transcarbamylase to form citrulline. Now citrulline will combine with aspartate in the presence of the enzyme argininosuccinate synthase 1 to form argininosuccinate, and this argininosuccinate will break down into arginine and fumarate by the enzyme argininosuccinase. This arginine will again break down into urea and ornithine in the presence of the enzyme arginase, and this ornithine will then enter the mitochondria so as to continue the cycle again. So urea, which is formed as a byproduct in the liver, will enter into the blood circulation. This urea will now be transported to the kidney where it is excreted in the urine. So urea is less toxic compared to ammonia, and it is more water soluble, so it is easily excreted in the urine. Now we will discuss urea cycle in more detail. Urea cycle was first described by Hans Krebs and Kurt Heinrich in 1932, and so it is also known as Krebs-Heinrich cycle. Urea cycle has 5 steps of cyclic process with 5 distinct enzymes. So urea cycle occurs in the liver in both mitochondria and cytoplasm. Out of 5 enzymes, first 2 enzymes are present in the mitochondria, that is carbamoyl phosphate synthetase type 1, that is CPS type 1, and the other one is ornithine transcarbamylase, that is OTC, and the rest of the 3 enzymes are present in the cytosol. In step 1, one molecule of ammonium combines with one molecule of carbon dioxide in the presence of the enzyme carbamoyl phosphate synthetase type 1, that is CPS type 1, to form carbamoyl phosphate. Now during this process 2 ATP is utilized and gets converted into 2 ADP. So phosphate present in the carbamoyl phosphate is derived from this ATP. Now since this enzyme carbamoyl phosphate synthetase type 1 is present in the mitochondria, so this reaction occurs in the mitochondria only, and N-acetyl glutamate is necessary for activation of this enzyme. So what are the properties of carbamoyl phosphate synthetase type 1? So this enzyme catalyzes the formation of carbamoyl phosphate from ammonia and carbon dioxide. This is a rate limiting enzyme of urea cycle. This is a rate limiting enzyme, and this enzyme is active only in the presence of N-acetyl glutamate and allosteric activator. So what happens when there is a defect in this enzyme? When there is a defect in carbamoyl phosphate synthetase type 1, then it will lead to a deficiency known as carbamoyl phosphate synthetase type 1 deficiency, and this deficiency is autosomal recessive disorder. And because N-acetyl glutamate is of utmost importance in the activation of carbamoyl phosphate synthetase type 1, in the defect of N-acetyl glutamate, it will result in the deficiency known as N-acetyl glutamate deficiency, which is autosomal recessive. Now coming to step 2 of this reaction. This reaction will also take place in mitochondria, where one molecule of carbamoyl phosphate condenses with one molecule of ornithine in the presence of the enzyme ornithine transcarbamylase, that is OTC, to get converted into citrulline. So carbamoyl phosphate that was synthesized from ammonia in step 1 combines with one molecule of ornithine, and the resultant compound formed is citrulline, and this citrulline will enter into the cytoplasm to lead the rest of the cycle. So what is the property of ornithine transcarbamylase enzyme? Ornithine transcarbamylase enzyme catalyzes the formation of citrulline from carbamoyl phosphate and ornithine, and when there is a defect in ornithine transcarbamylase enzyme, it leads to a condition known as ornithine transcarbamylase deficiency, which is an X-linked disorder. This is an X-linked disorder. Now coming to the third step of urea cycle. After citrulline moves out of mitochondria into the cytoplasm, it gets condensed with one molecule of aspartate in the presence of the enzyme argininosuccinate synthetase type 1 to form argininosuccinate. Because it is synthesizing argininosuccinate, the enzyme is argininosuccinate synthetase type 1. And during the reaction, ATP is converted into AMP and there will be release of inorganic phosphate. The property of argininosuccinate synthetase type 1 is it is a ligase enzyme and it links amino nitrogen of aspartate to citrulline to form argininosuccinate. And when there is a defect in argininosuccinate synthase type 1 enzyme, then the condition is known as citrullinemia type 1 and it is a autosomal recessive disorder. Now coming to step 4 of urea cycle, argininosuccinate is broken into two molecules, that is fumarate and arginine, in the presence of the enzyme argininosuccinate lysase. So argininosuccinate lysase, it cleaves or breaks down argininosuccinate into fumarate and arginine. And when there is defect in argininosuccinate lysase, there will be accumulation of argininosuccinate and this will result in argininosuccinic acidemia. And in this defect, we will see a characteristic feature of trichorexis nodosa, that is, there will be dry and brittle hair. In this step, arginine will again break down into urea and ornithine in the presence of the enzyme arginase, and during the process one molecule of water is introduced in the reaction. So this enzyme arginase, it is a hydrolase enzyme. It is a hydrolase enzyme and it causes hydrolytic cleavage of arginine to release urea and ornithine, and this ornithine will re-enter the mitochondria. Now when there is a defect in arginase, it leads to a condition known as Arginase Deficiency, which is a autosomal recessive disorder. And the characteristic feature of Arginase Deficiency are it leads to spastic tetraparesis, microcephaly, intellectual disability and mild hyperammonemia. And during the process, the urea formed will be taken into the circulation and will reach the kidney. And since it is water soluble, it easily gets excreted in the urine. There can also be a defect in mitochondrial ornithine transporter type 1. And this condition is known as hyperornithinemia, hyperammonemia, homocitrullinuria, and that is known as HHH syndrome. This is also an autosomal recessive disorder. So we saw that all the defects in the urea cycle was autosomal recessive disorder except ornithine transcarbamylase deficiency, which was X-linked disorder. Now this hyperornithinemia, hyperammonemia and homocitrullinuria, that is HHS syndrome, will present with vomiting, lethargy, failure to thrive, episodic confusion, hyperammonemia and protein intolerance. And there can also be defect in mitochondrial aspartate or glutamate carrier, that is CTLN2. And this condition is known as citrullinemia type 2, which is also a autosomal recessive disorder. And this citrullinemia type 2 will present with neonatal intrahepatic cholestasis in neonates, and in adult it presents with sudden behavioral change and stupor, and they may present with coma and hyperammonemia. Points to remember in urea cycle. This is important for MCQs. So compound consumed in urea cycle are carbon dioxide, ammonium and aspartate. Compound that enters into urea cycle and is regenerated again is ornithine. So this is also known as ornithine cycle. Two ATPs are used in formation of carbamoyl phosphate and two ATPs are used in formation of argininosuccinate. So in total in urea cycle there are 4 ATPs that are consumed. Now overall reaction of urea cycle can be summarized as 1 molecule of ammonia, 1 molecule of carbon dioxide and 1 molecule of aspartate will give you 1 molecule of urea and fumarate as a byproduct. So aspartate is consumed, fumarate is a byproduct, and there is no gain or loss of ornithine, citrulline, argininosuccinate and arginine. And the source of nitrogen atom of urea are one nitrogen atom comes from ammonia and the other nitrogen atom comes from amino group of aspartate. Urea is also linked to TCA cycle through fumarate and aspartate, and hence this is also known as urea bicycle. Now how will you treat this patient? Treatment includes removal of ammonia by nitrogen scavenger. So these drugs are sodium benzoate, sodium phenyl acetate, sodium phenyl butyrate and glycerol phenyl butyrate. So these drugs divert nitrogen away from the urea cycle by promoting the synthesis of nitrogen rich metabolites which are excreted at high rates in the urine. And we can also give arginine supplements, and these arginine supplements will provide more ornithine so that ammonia will be metabolized to form urea and which will finally get excreted in the urine. So this was overview of urea cycle. I hope you got the concept of urea cycle. Thank you.